Hello and welcome to the first instructional video on photosynthesis. I'm sure most of us have learned the concept of photosynthesis, but trust me, there is much more. First, let's start with the reactants. In photosynthesis, carbon dioxide, water, and light energy are the starting components of this process. With the help of chlorophyll, the reactants can become the products: glucose, oxygen, and water. The next question would be how a plant can get the chlorophyll needed to produce glucose and oxygen. Well, the answer is right in a plant cell, the chloroplast. The chloroplast contains chlorophyll and has a double membrane. Every chloroplast has stacks of thylakoids. These stacks are called granums. The sole purpose of a thylakoid is to simply absorb light. With the light energy, water can be reorganized to form. The next key component in a chloroplast is the stroma, which would be the liquid surrounding the thylakoid. The stroma is where the dark reactions occur. These dark reactions have three names: the Calvin cycle, C3 photosynthesis, and carbon fixation. All of these three names mean the same thing. CO2 is changed to glucose. The energy source that dark reactions use are ATP and NADPH. Let's go back to the thylakoid, where the light reactions happen. The light reaction energy is simply light from the sun. Light is electromagnetic with properties of both a particle and a wave. Wavelength of light impacts the color of light. Wavelengths from 380 nanometers to 750 nanometers are visible light. A greater wavelength results in less energy, and a smaller wavelength will result in more energy. Our sun emits all wavelengths of light, but our atmosphere usually only allows visible light to come through. This is important to photosynthesis because of pigments. Pigments are chemicals in plant cells that absorb some wavelengths of light and reflect others. The light that it reflects is actually the determining color of an object. Take an apple, for example. This apple is red, which means it is reflecting red light. Plants have three pigments that are essential to the completion of photosynthesis. Chlorophyll A is the main component of photosynthesis. It reflects green light while mostly absorbing blue and red light. Chlorophyll B is another pigment in plants that reflects yellowish green light while mostly absorbing light blue and orange light. Carotenoids reflect yellowish orange light while mostly absorbing light blue and dark green light. Now, let's take a closer look at what actually happens in the thylakoid. Each thylakoid has two photosystems. Photosystem 1 can work independently, and it is where 700 nanometer light is absorbed, which is why it also goes by P700. P700's electron transport chain creates NADPH. Photosystem 2 absorbs 680 nanometer wavelengths of light and is dependent on P700's presence. Because of the light it absorbs, it can be called P680. P680's electron transport chain creates ATP. In both photosystems, there is an antenna complex. The antenna complex is composed of multiple pigments, including chlorophyll B and carotenoids that gather light energy and move it between particles. The reaction center is composed of chlorophyll A. This is where all of the absorbed light energy from the antenna complex is dumped into. In this center, chlorophyll A transfers an excited electron. This excited electron is trapped by the primary electron acceptor, which then sends the electron through an electron transport chain. In the thylakoid membrane, electrons pass through water. This system is called non-cyclic electron flow. This process produces NADPH, ATP, and oxygen. Chemiosmosis also happens in the thylakoid. This is the creation of positive hydrogen ions. Electron transport allows for energy to be made so that the hydrogen ions can be moved to the inner thylakoid. When these ions build up in the thylakoid, ATP synthase allows for the hydrogen ions to move out of the cell. This is because particles naturally move from a high concentration to a low concentration. Another very important process happens in the thylakoid once light is absorbed: photophosphorylation. Photophosphorylation is the process of adding a phosphate to ADP. With the added phosphate, ATP is produced. There are two types of photophosphorylation: non-cyclic and cyclic. Non-cyclic photophosphorylation is when electrons are not cycled but go in one direction to create NADPH. Cyclic photophosphorylation is when electrons are cycled through a system, creating ATP. Usually this occurs in photosystem 1. The produced ATP and NADPH from the light reactions goes on to fill the energy source that is needed in dark reactions in order to be successful. Photophosphorylation creates an important law in photosynthesis. Dark reactions will not happen unless light reactions happen first. Now that we've talked about light reactions, let's discuss what happens in dark reactions. First, we'll start with the names. Dark reactions have three names because of three different aspects. The Calvin cycle comes from the scientist who first researched it, Melvin Calvin. C3 photosynthesis comes from the fact that 3 carbon compounds are created in the process. And finally, carbon fixation comes from the fact that CO2 is added to make sugar. For simplicity, I will refer to the dark reactions as the Calvin cycle. The Calvin cycle has 4 main stages. It starts off with carbon fixation, then reduction, then making the sugar, and finally RUBP regeneration. In carbon fixation, there is a molecule called ribulose bisphosphate, or RUBP for short. RUBP is a 5-carbon sugar with 2 phosphates. CO2 binds to the RUBP through the help of an enzyme called RUBP carboxylase, or Rubisco for short. Rubisco happens to be the most abundant enzyme on Earth. It takes in carbon dioxide and binds it to RUBP. This forms an unstable 6-carbon compound. This compound then breaks down into 2 3-carbon compounds that only have 1 phosphate. These compounds are called 3-phosphoglycerate, also known as PGA. With the use of ATP and NADPH from the light reactions, PGA is changed into glyceraldehyde 3-phosphate, which can go by the name of PGAL or G3P. One G3P is released from the cycle and the rest of the G3P is rearranged with the help of ATP to form more RUBP. This RUBP is reused in the cycle and another G3P molecule is released. The 2 G3P molecules that were released are rearranged to form glucose. The production of glucose can decrease due to the process of photorespiration. This occurs when Rubisco cannot distinguish between oxygen and carbon dioxide. Researchers believe that Rubisco evolved before there was more free oxygen available in the atmosphere, so there was no need to distinguish CO2 and oxygen. When it is hot outside, the stoma closes to preserve water. Stoma are the pores under a leaf that take in carbon dioxide and release oxygen. Naturally, oxygen builds up in the leaf because there is no way out. This results in Rubisco accepting oxygen instead of carbon dioxide. So, there is no room for carbon dioxide to be accepted into the Rubisco. Without carbon dioxide, PGA cannot be formed, which delays the production of glucose. Also, in dark reactions, some plants go through C4 photosynthesis. Instead of RUBP, PEP is used. During C4 photosynthesis, a 4-carbon compound is created instead of a 3-carbon compound. In C4 photosynthesis, 2 cells are in use. There are bundle sheath cells and mesophyll cells. Bundle sheath cells are the place where the Calvin cycle occurs and are tightly packed around leaf veins. In these cells, thylakoids are not stacked. Mesophyll cells are loosely arranged between the bundle sheath cells. These cells take in CO2, form a 4-carbon compound with PEP, and dump the compound into the bundle sheath cells. Once inside, the 4-carbon compound breaks down into a 3-carbon compound and CO2. An advantage of using C4 photosynthesis would be that the bundle sheath cells are flooded with carbon dioxide. So, it is more likely for rubisco to accept CO2 more often than oxygen. This reduces the occurrence of photorespiration, which in turn will make the reduction of glucose rarely occur. Desert plants use a version of photosynthesis called crassulacean acid metabolism, or CAM for short. In CAM, the stoma opens at nighttime to take in CO2 and release oxygen. The absorbed CO2 is incorporated into organic acids that release CO2 during the day so that the stoma can stay closed and reduce the chance of oxygen being accepted by rubisco. Although very similar, CAM and C4 photosynthesis are different. C4 photosynthesis uses cells to separate the processes of carbon fixation, while CAM uses time to separate the processes of carbon fixation.